Our next speaker is Kerry Geiler-Samerotte.
Thank you. That's the closest anyone's got to pronouncing my name correctly. I'm Kerry Geiler-Samerotte, and I'm going to be talking to you today about a question. And that question is whether selection transforms the patterns of epistasis we see in nature. So we know that selection acts as a filter of new mutations. For example, eliminating new mutations that are strongly deleterious, such that the genetic variants left in nature represent a biased subset of what's possible. So my question is, does selection also filter the types of epistasis that are present in nature? And in particular, I'm interested in a protein-folding chaperone called Hsp90. It interacts epistatically with a lot of the genetic variants in nature, and it does so in a very specific way, and that's that it tends to buffer their effects. So the first time this was observed was in an experiment done in fruit flies. And in this experiment, they took a bunch of lines of genetically distinct flies and inhibited Hsp90. And when they did so, they saw that the previously buffered effects of cryptic genetic variation are revealed. And you see a different phenotype is revealed in each fly line. That's because these fly lines are genetically distinct. So each one possessed different buffered alleles. This effect has been repeated in different model organisms, in flies, in plants, in fish. So I don't think there's much of a question anymore as to whether Hsp90 tends to buffer the genetic variation in nature. But there is a big question about why. And one hypothesis as to why is that selection transforms the types of epistasis that we find in nature. So in this hypothesis, Hsp90, it might buffer the effects of some new mutations, but it might interact with other new mutations in a completely different way, maybe enhancing some of their effects. And it's selection that comes in and preferentially allows buffered alleles to persist through evolutionary time, leaving this false impression that Hsp90 predominantly acts as a buffer. But there's another more straightforward hypothesis as to why Hsp90 tends to buffer the effects of genetic variation found in nature, and that's that Hsp90 it just is a buffer. It buffers genetic variation in nature, it buffers the effects of new mutations. We call this the robustness hypothesis because it means that, if it's true, it means that Hsp90 makes genomes more robust to the effects of new mutation. To distinguish between the selection and the robustness hypothesis, I'm going to study how Hsp90 interacts with new mutations and whether it tends to buffer their effects. So I do this study in yeast, and I focus on two collections of yeast strains that accumulated new mutations or new recombinations under reduced selection pressure. And as a control, I also studied two collections of yeast strains isolated from nature. The first I acquired from different breweries, and the second is a published yeast collection of yeast from different environments around the world. So the phenotypes that I study are single-cell morphologies. So I'm worried that the mouse or the fly people are sort of laughing, like, single cells don't have morphology, but they do. They have some interesting phenotypes that are related to the way they grow. You can measure things like cell size or bud angle, the position of the nucleus. I use high-throughput microscopy to measure 200 different morphological features. Ultimately, I studied 29 principal components that each explain greater than 1% of the variation in single-cell morphology. And the way we quantify the effect of Hsp90 is similar to what previous studies have done. We inhibit Hsp90 and see if phenotypic variation is revealed between strains. And the way we inhibit Hsp90 is with a drug called galdanomycin. It's administered at low levels, levels that have a minimal effect on the growth rate of yeast cells. So the first result I want to show you is that we can confirm the results of previous experiments showing that Hsp90 tends to buffer genetic variation in nature. So in this plot, each point represents the phenotype of a single yeast strain that was isolated from a brewery, and this blue bar represents the amount of phenotypic variation between those strains. And when I inhibit Hsp90, you can see that the amount of variation increases, suggesting that Hsp90 was acting to buffer that variation. This is only one principal component. So now let's look across all 29 principal components in all 4 collections of yeast strains. the 2 collections that harbor standing genetic variation, and the 2 collections that harbor either new mutations or new recombinations. Every point in this plot represents, for a single principal component, the change in phenotypic variation upon Hsp90 inhibition. So more simply put, this point represents the height of that red bar minus the height of the blue bar in the plot to the right. And for this particular principal component, Hsp90 increases the amount of phenotypic variation, so this point is to the right of the 0 line. So now let's look at the other 29 principal components. And you can see that a few of them also have significant increases in variation upon Hsp90 inhibition. But moreover, the distribution across all 29 principal components has a median that's significantly greater than 0. So this suggests that in this collection of yeast strains and in the other collection of yeast strains isolated from nature, Hsp90 does tend to buffer the effects of genetic variation. So what about new mutations? So let's look at the other yeast lines. And here we actually see the opposite effect. So when Hsp90 is inhibited, phenotypic variation between these strains shrinks. And what this suggests is that when Hsp90 was present, it was enhancing the effects of new mutations, such that when you take it away, the yeast lines become more similar to one another. So let's look at an individual principal component just to understand this better. So we'll look at this principal component here, and you can see that for this PC, the yeast lines in the control condition have greater phenotypic variation between them than in the Hsp90 inhibited condition. And this is exactly the opposite of what we saw for the same PC in the brewing strains. So this contrast between what we see in yeast strains harboring genetic variation that has survived selection versus yeast strains possessing genetic variation that has not necessarily been screened by as strong of a selection pressure, this contrast is the first piece of evidence in favor of the selection hypothesis, telling us that selection transforms the types of epistasis found in nature. Another piece of evidence comes from if we look at principal components that don't appear to be strongly affected by Hsp90 inhibition. So here's such a PC, and you can see that when we inhibit Hsp90, the amount of phenotypic variation between these strains doesn't increase too much or decrease, but you can still ask, is Hsp90 inhibition having an effect on individual yeast strains? So I'll show you that by drawing a line connecting the point representing the phenotype of a yeast strain in the control condition to its phenotype in the inhibited condition. And you can see not all of these lines are horizontal, so Hsp90 has an effect on the phenotype of some of these individual yeast strains. The strains that are most affected differ depending on what principal component we're looking at. Now, this collection of yeast strains I'm showing you, these are the MA lines. So these are the lines that possess new mutations that haven't been filtered by natural selection. So what these results suggest is that Hsp90 interacts with a lot of new mutations, and it does so in diverse ways. It buffers the effects of some of them. So for example, the blue line, MA32, probably possesses a mutation that's effect are buffered by Hsp90. But other lines like MA line 42, the yellow line, Hsp90 seems to be enhancing the phenotypic effect of mutations in those lines. And this is another piece of evidence in favor of the selection hypothesis, because what it basically says is that Hsp90 interacts with new mutations in diverse ways, and it suggests it's selection that's preferentially enriching for buffered alleles in nature. So to conclude, the main message of this talk is that selection transforms the types of epistasis that we find in nature. You might be thinking, okay, well, what does this mean about Hsp90 and other potential buffers? So on one hand, I think it means that Hsp90 is not, strictly speaking, a buffer. It does not buffer the effects of new mutations. It does not tend to. And this calls into question whether other gene products, any gene products, can increase robustness to the effects of mutation, or whether it's always selection that's transforming patterns of epistasis and kind of leaving this false impression of robustness in genomes. On the other hand, even if Hsp90 is not a buffer, it's still pretty interesting. It interacts with a lot of new mutations, modifying their effects. It interacts with standing genetic variation. We recently asked whether it also interacts with adaptive mutations and found that it did. In this plot, every point represents a yeast strain with a single adaptive mutation in one of these 3 genes. And you can see that Hsp90 inhibition modifies the fitness advantage of these lines in a gene-dependent way. Depends on what mutation or what gene that mutation is in. So to conclude, Hsp90 may not be a buffer, but I think it's still a really interesting protein. And it's interesting to think about how mildly perturbing the effects of just this one protein can affect a lot of genetic variation and may have a big impact on evolutionary processes. So to conclude, I want to thank my postdoc advisor, Mark Siegel, and the Siegel Lab at New York University. That's where most of this work was done. I also want to thank the team that constructed the MA lines and that sequenced them so that we could study them. And finally, I want to thank the Petrov Lab at Stanford University and our collaborators there. That's where we've been pushing this work forward, studying how Hsp90 interacts with adaptive mutations. And I also want to thank all of you. I'll take questions.
Thanks for the talk. I have perhaps a naive question. Have you thought about possibility that maybe particularly for the MA lines, the difference arises from the different lines have different sensitivity to the drug you administered? And suppose the Hsp90 have some sort of optimum concentration in the cell, too much or too little will reduce or sort of exacerbate some gain function mutations. But I guess I'm trying to say maybe the MA line result doesn't specifically show the interaction between the new mutation and the Hsp90, but rather maybe some sensitivity to the drug you administered.
Yeah, and I understand your question. It's about whether different MA lines have different sensitivities to the drug, and that explains the pattern. So each MA line has a very small number of new mutations, on average 4, and the drug concentration I'm using is low. So I don't think that's what's happening, but I guess I was thinking about the same question a different way, like maybe the ancestor of the MA lines has some different interaction with Hsp90, and that's why we saw this effect that's opposite of what we see in nature. And to contend with that, that's why we also studied the rec lines, to see if we can get another independent collection of strains that harbor variation that hasn't been exposed to selection and see if we'd see the same thing, and we did. So I think that has more to do with mutations or being filtered or unfiltered by selection than sensitivity to the drug.
Thanks.
Do you think Hsp90 is interacting directly with these proteins encoded by the new mutations, or do you think it's indirect?
I'm super glad you asked that. So I think that so I don't know, but I think that it's probably indirect because the buffering hypothesis makes a lot of sense to me if it's a direct interaction.
Exactly.
You have this protein, it has a mutation that hurts its ability to fold, Hsp90 helps it to fold, fixes the problem, buffers it. But if you zoom out and realize that every interaction in the cell isn't just a 2-by-2 interaction, you have these long pathways, and maybe Hsp90 activates one of these pathways, and it helps an activator of the pathway to fold. Imagine if you have mutations sort of downstream. If you inhibit Hsp90 and you turn the pathway off or shut it down a little bit, you're going to reduce the effects of those mutations. So if you have Hsp90 functioning, it's enhancing the effects of mutations. And maybe that explains why it tends to enhance the effects of new mutations, but that's not something I've studied. It's just one idea to explain this kind of unexpected result.
Is there any chance that Hsp90, when not exposed to selection for a long time, loses its buffering ability? And that the HSP alleles that are in the MA lines
Sorry, can you speak up a little bit?
Sorry. Is there any chance that Hsp90 alleles that have spent a lot of time without selection sort of lose their buffering capacity, and that your MA line Hsp90 alleles work differently than Hsp90 alleles that you would find in the wild?
So you mean that Hsp90 itself is mutated in the MA lines? Is that the question?
Yes, that's the question.
So these lines have been sequenced, so you can see where the mutations are, and there aren't any in Hsp90. But that's related to the first question, or to this fear that maybe the ancestor of the MA lines have something different going on with their chaperone system. That's why we studied a separate collection of yeast lines.
Yeah, that's really cool. Thank you.
So I know those MA lines have a pretty high frequency of beneficial mutations, allegedly. Did you try to separate them out and see if you see a similar pattern with just the deleterious lines?
I'm sorry, can you speak up again?
Yeah,
The
so,
MA
so
lines?
those MA lines are claimed to have a pretty high frequency of beneficial mutations. So if you cull those out, do you see a similar result?
The MA lines,
Yeah, so it is alleged that those lines have a relatively high frequency of beneficial mutations.
oh, higher than expected? I'm not
Well,
sure. I
so-
can take a look. I have looked at the severity of the mutations, so whether they're synonymous or in protein coding genes, or whether they're a mutation in a protein coding gene predicted to disrupt protein function. And there you do see, actually, it's right there. There you do see the mutations with a more severe effect on protein function seem more likely to interact with Hsp90, basically. Thank you.